Hello and good afternoon, and thank you for joining this special Alcyomics webinar in association with the DSTC, Drug Safety Testing Center, Japan. I'm Asif Tula, and I'm the Business Development Manager for Alcyomics. Today, I'm going to talk you through how Alcyomics provide novel safety testing solutions to the pharmaceutical industry using our patented Skimmune assays, which uses fresh human skin and fresh human blood. If you have any questions throughout this webinar, please type them in the message bar on the webinar platform. Alternatively, if you have any questions after the webinar is completed, please feel free to send those to me via my email. I'll start by giving an introduction to Alcyomics before giving you a background introduction to the technology Skimmune. I'll then describe how Skimmune can be used in the pharmaceutical industry, focusing on small molecule drugs and biologics, before introducing a new development, Skimmune 3D, our full thickness 3D skin equivalent model, and how this can be used as a screening tool to determine immunogenicity to biologics. So firstly, an introduction to Alcyomics. Alcyomics is a biotechnology company based in the northeast of England in Newcastle upon Tyne. We have been trading since 2009 and offer a unique service provision based on preclinical product safety testing. Our tests are based on human in vitro skin tests, which we call Skimmune, and Skimmune has been developed on more than 20 years' experience of using the skin explant model in a research setting. Skimmune is a non-animal alternative model and supports the 3 Rs, the reduction, refinement, and replacement of animals in research. So Alcyomics have developed Skimmune as immune-mediated drug hypersensitivity is a cause for concern in the industry. Around 20% of drugs which enter clinical trials fail due to safety, and around 10% of these are a result of immune-mediated drug hypersensitivity reactions. Predicting which new drugs can cause these immune-mediated drug hypersensitivity reactions is important, particularly as most preclinical studies are conducted in animals. One such example of where this can go wrong was the Northwick Park trial disaster with the T. Gennaro antibody, which occurred in London in 2006. In this trial, all the subjects suffered severe organ dysfunction as a result of cytokine release syndrome. This was not predicted based on the preclinical animal studies and highlights the importance of the need to develop reliable human-based models to assess drug safety. Alcyomics therefore developed Skimmune to help our clients identify adverse drug reactions before they take their product into the clinic. We can help you reduce risk and cost in the drug development program by the early identification of potential adverse reactions. Skimmune is a unique human in vitro model, which we have shown through published data to be highly predictive of clinical classification of adverse reactions. And Skimmune is also applicable to different drug types, including small molecule drugs, biologics, including monoclonal antibodies, their aggregated format, biosimilars and biobetters, nanomedicines, oligonucleotide therapeutics, as well as cellular therapies. So I wanted to go on now to give you a bit of background to the technology. Skimmune is based on the skin explant assay, which was originally developed by Vogelsang et al. In 1985. It is an in vitro predictive test for graft versus host disease in patients with genotype HLA identical bone marrow transplants. Graft versus host disease is a major cause of morbidity and mortality in bone marrow transplant recipients. Donated bone marrow or peripheral blood stem cells view the recipient's body as foreign and attack, which causes a cytokine storm. It can be used as a tool for selection of donors for transplantation and prediction of transplant rejection and graft versus host disease. We can use the skin explant model to predict graft versus host disease. We can start with a mixed lymphocytes culture. We can start with a mixed lymphocyte culture with irradiated transplant lymphocytes and HLA-matched donor lymphocytes. The skin explant culture involves a 4 mm punch biopsy, which is taken from the patient and further excised into smaller skin explants before being cultured with the sensitized donor lymphocytes for 3 days in a co-culture. After 3 days, the skin sections are harvested, fixed, and paraffin embedded before histopathological grading of the skin sections for damage using the Lerner classification. So I'll now go on to give you an introduction to the SCIMMUNE assays. The SCIMMUNE platform predicts hypersensitivity responses to pharmaceuticals, including small molecule pharmaceuticals and monoclonal antibodies, chemicals, and cosmetics. This is a human in vitro test, and we have access to fresh human skin and fresh human blood from the same individual. This next slide shows you the assessment of small molecule drugs using the SCIMMUNE platform. We obtain skin and blood from a healthy volunteer. We extract the immune cells from the blood to make monocyte-derived dendritic cells and allow the test compound or the test drug to present itself to the immune system by interaction with the MODCs. These monocyte-derived dendritic cells are then co-cultured with autologous T cells. If the test article or test drug activates the immune system, the T cells will be primed such that when co-cultured with the skin from the same individual, the T cells will release cytokines which cause damage in the dermal epidermal junction of the skin. This damage can be graded clinically using a clinical grading classification called the Lerner classification. SCIMMUNE assays are ideally suited to the assessment of adverse immune reactions to small molecule drugs and protein molecules, and the assays can be used to determine hypersensitivity reactions and ensure drug safety. For the assessment of small molecule drugs, Alcyomics also provides screening assays for the early identification of adverse events. These screening assays are useful for screening compounds quickly. However, due to the variation between donors, they can be difficult to identify compounds as problem or not. This is due to the error bars spanning the cutoff line. While our screening assays require less donors to identify problem compounds or not, when compared to other assays on the market, the use of SCIMMUNE, due to the addition of the skin tissue and the histopathological readout, makes it more easy to identify compounds as sensitizer or non-sensitizer. This is due to the specificity of the response causing damage to the skin tissue. The assay for the assessment of monoclonal antibodies and biologics is slightly different in that the peripheral blood mononuclear cells, PBMCs, the skin, and the test article are all cultured at the same time. However, the histopathological readout is the same. The assessment of adverse immune reactions to monoclonal antibodies, biosimilars, and biobetters can be used with Skimmune Mab. It's a first-in-man test and offers a preclinical assessment of adverse effects in the clinic. It's a human in vitro test, so there are no interspecies differences or difficulties in data interpretation, and it's a preclinical prediction which offers the early identification of potential adverse events and prevents costly failed clinical trials. This slide gives an example of how we grade the histopathological damage. We can grade the damage using the learner classification, which is a clinical system used in clinical practice in the past, and so is validated. The damage goes from grade 1 on the left to grade 4 on the right. Grade 1 is a negative response, and this is normal, healthy skin. There is no damage to the skin tissue. Grade 2 is where we start to see a positive response, and we can see this by the formation of small vacuoles in the dermal-epidermal junction. This would present as a rash in a clinical trial. With grade 3, the damage is more severe, and we see the vacuoles merging to form larger clefts. We also see necrosis in the epidermal regions. This would present as blistering in the clinic. And grade 4, which is the most severe response, is where we see complete separation of the proteins and the connected markers in the dermis and the epidermis. This was like the top layer of the skin coming off. In Alcyomics, the grading process is performed blind by 2 independent histopathologists who are trained in this grading process. We developed Skimmune. When we developed Skimmune, the first thing we did was to validate the technology against the gold standard mouse local lymph node assay, LLNA. We tested 42 chemicals, 21 were known as sensitizers and positive compounds, and 21 as negative compounds. We ran these through the Skimmune assay in n=10 donors, and we found that Skimmune could correctly identify those as sensitizer or non-sensitizer with a 95% specificity, 95% sensitivity, and accordance rate of 95%. This work was published in 2015. We have developed our Skimmune assay to bridge the gap between preclinical animal studies and first-in-man human clinical trials. Skimmune represents a human in vitro test that has been shown to predict the response of drugs with a high degree of predictability to their clinical classification. So this quite nicely sets you up before the designing of clinical trials. I will now go on to describe some of the examples of using the Skimmune assay in the pharmaceutical industry. Once again, this is a schematic of how the Skimmune assay works for the immunotoxicity assessment of small molecule drugs. We have skin and blood from a healthy volunteer. We extract the relevant immune cells from the blood to make monocyte-derived dendritic cells and allow the test compound or the test drug to present itself to the immune system by interaction with these dendritic cells. The dendritic cells are then co-cultured with the T cells. If the test article activates the immune system, the T cells will be primed such that when co-cultured with the skin from the same individual, the release of cytokines will cause damage to the skin at the dermal-epidermal junction. This slide shows some work that we recently completed with Pfizer, and this was published in March last year. In a blinded study, we were given 17 small molecule drugs which are used in clinical practice. We did not know whether these were positive drugs or negative drugs, and we tested these in the Skimmune assay in n=10 donors per compound. using the clinical dose. From our studies, we were able to correctly identify those drugs which were positive and those drugs which were negative, but we were also able to show with a high degree of correlation whether the drugs were likely to cause a frequent adverse drug reaction, a common adverse drug reaction, or a rare event. We showed that the percentage of positive tests in Skimmune correctly correlated with the percentage of clinical classification of adverse drug reaction. This table shows the data in more detail. Here are the 17 drugs that were tested, and you can see that the table shows that the percentage of positive tests in Skimmune directly correlates with the percentage of clinical classification of adverse drug reaction. This was a really nice study for Alcyomics, and as mentioned, this was published in March last year. I will now go on to describe some of the examples of using Skimmune in the biopharmaceutical industry. The number of biologic drugs being developed has increased exponentially over the last few decades, and they have been developed for many different diseases or therapeutic indications. The following table describes some of the differences between small molecule drugs and biologics. Biologics are much larger when compared to small molecule drugs and are also more complex as they are produced in living cells. Being produced in living cells makes them very heterogenic and difficult to characterize. They are also costly to produce and are very immunogenic, so they can promote an unwanted immune response in an organism. At Alcyomics, we have developed the Skimmune MAB assay for the assessment of biologics, including monoclonal antibodies, biobetters, and biosimilars. In the Skimmune MAB assay, the test biologic, the PBMCs, and the autologous skin are co-cultured at the same time. There is no DC activation step, which we use when assessing small molecule drugs. We have shown in our previous work during a retrospective study that Skimmune MAB could have predicted the life-threatening events caused in the T. Janeiro monoclonal antibody test at Northwick Park. As you can see from this image, Skimmune MAB would have predicted a grade 3 positive response as a result of the damage in the dermal epidermal junction highlighted by the black arrows on the diagram. If Skimmune MAB was available at the time, we would have advised not to conduct the clinical trial with the current monoclonal antibody. We have also assessed many monoclonal antibodies which are currently used in clinical practice using Skimmune MAB, and we have shown concordance with our small molecule data in that Skimmune MAB can correctly identify those monoclonal antibodies which are positive or not by causing hypersensitivity reactions. Our data also show that the percentage of positive reactions in Skimmune MAB correctly correlates with the frequency of hypersensitivity reaction observed in the clinic. For example, OKT3 and TGN1412 are correctly identified as extreme sensitizers, and this is what is observed in the clinic. Therefore, using Skimmune MAB to test your biologic under development will provide you with a high degree of likelihood of what response you will see in a human trial, and so can be used to predict whether you would likely see problems in a first-in-man study. Interestingly, the highlighted row here represents an unexpected response, as Humira should be a negative antibody, but this has come up as positive in Skimmune with 70% positive tests. Further analysis by our scientists indicated that this monoclonal antibody was actually an aggregated preparation, and so this positive response is what we would expect as aggregated monoclonal antibodies are known to cause adverse reactions. This data shows the correlation data between positive responses observed in the clinic and a positive classification in the SCIMMUNE MAB assay. I will now describe some case studies where I will show you how we have worked with our clients in the past. The following case study is looking at the assessment of monoclonal antibodies. In this particular study, a client provided Alcyomics with 3 preparations. Preparation 1 contained a monoclonal antibody which failed a phase 1 clinical trial. Preparation 2 was a reformulated version of this failed antibody, and preparation 3 was a negative control. You can see from the data that antibody B is the antibody which failed the clinical trial. We can see this as a result of grade 2 damage in the dermal epidermal junction and also necrosis in the epidermal regions of the skin tissue. Antibody A was actually the reformulated antibody, and from this slide we can see grade 1 negative damage. Therefore, this reformulated antibody would be okay to progress onto clinical trials. Antibody C was the negative control, and in this we also see a grade 1 negative response. I mentioned previously about aggregation to monoclonal antibodies. Aggregation is a critical quality parameter during the manufacture of monoclonal antibodies, and aggregated peptides are not normally detected using less sensitive blood-based assays such as T-cell proliferation and cytokine release assays. SCIMMUNE MAB with the histopathological evaluation is more sensitive to predict this output. The data here shows aggregated preparations of Herceptin and rituximab showing a grade 2 positive response or above in response to high temperature or high pH, which causes aggregated preparations. The next case study that I wanted to describe to you is toxic epidermal necrosis, otherwise known as TEN. This slide shows a different type of damage to what we have seen before. With the SCIMMUNE assay, we have previously been looking at damage at the dermal epidermal junction. This is typical graft versus host disease type damage. However, SCIMMUNE MAB has been shown to detect a different type of damage in the upper epidermal regions of the skin tissue. This damage represents toxic epidermal necrosis and has been observed in some of our studies when working with monoclonal antibodies. This shows an additional use of the SCIMMUNE assay to detect a TEN-like damage profile. This information can therefore be used by product developers to reformulate or redesign the monoclonal antibody to ensure that this type of damage doesn't occur. Of course, identifying this before a clinical trial is particularly advantageous. We can also use the assay to look at the efficacy of immunomodulatory peptides and proteins by using the assay in a slightly different way. In this form, we start with a grade 3 positive response, which can be induced by a mixed lymphocyte reaction. We can then add the test monoclonal antibody at different concentrations with the aim of trying to reduce a grade 3 positive response to a grade 1 negative response. You can see in this example that testing the monoclonal antibody at 0.15 microgram per mil reduces the grade to a grade 2 positive response, but a higher concentration of 1.5 micrograms per mil reduces the histopathological damage to a grade 1 negative response. And so this is the most optimal concentration that should be used with this particular monoclonal antibody. I have also mentioned that the SCIMMUNE MAB assay is suitable for the assessment of biosimilars and biobetters. In this example, a biosimilar highlighted as X was tested or compared to its reference commercial product highlighted Y. You can see for this data that the biosimilar X is actually more toxic than its reference product, as we can see a grade positive response when testing this compound compared to the reference product. This data was also shown in the T cell proliferation and interferon gamma results. The next thing that I wanted to talk about was the use of Scommune in the pharmaceutical industry, but the use of Scommune 3D. Alcyomics has developed Scommune 3D as a full-thickness 3D skin equivalent model, which is bioprinted in a 96 well plate format. We have currently designed this assay for the quick screening and safety assessment of biologics in a medium throughput manner. We have shown Scommune 3D to be representative of normal human skin at the protein and the mRNA level. You can see from this slide here that Scommune 3D on the bottom is compared to normal human skin at the top. We see the correct markers of extracellular matrix production and dermal formation, such as collagen 3, showed here. We also see structural keratins in the lower epidermis, such as keratin 14, and we get terminal differentiation of the epidermis, shown by the marker involucrin, shown here. Scommune 3D can be used as a suitable assessment for the safety assessment of monoclonal antibodies. In this particular study here, we showed how we can test a known positive and negative control monoclonal antibody using Scommune 3D. In this study, the positive antibody shows the expression of heat shock protein 70 in the epidermis. This is a marker of apoptosis and so indicates adverse reactions caused by the monoclonal antibody. The negative antibody and the standard controls do not show the presence of heat shock protein 70. Interestingly, this data also correlates with cytokine release, where the positive antibody OKT3 has increased levels of interferon gamma and TNF alpha when compared to the negative antibody and the standard controls. These are the expected results and show how Scommune 3D mAb can be used for the safety assessment of monoclonal antibodies, providing two outputs, one at the histopathological level and the other at the cytokine level. As mentioned previously, at Alcyomics, Scommune 3D is made by bioprinting cells into a 96 well plate format. This is just a slide showing our custom-made bioprinter, which we use to print the dermal fibroblasts before the printing of the epidermal keratinocytes on top and allowing the skin model to terminally differentiate before using in our assays. So to finish off, I just wanted to describe to you how at Alcyomics we work with our clients. We can work with you in a standard way, which initially involves working under a non-disclosure agreement, and we normally start by initiating a telephone call or a conference call to scope your initial needs. Our scientific team will provide their expert advice, and with you, we will agree an outline for a detailed project plan before issuing you with a proposal. Our projects are milestone-based, and this is initiated with you under regular client updates. The milestones are clearly set out and are continually reviewed. The final stage is to report back the study to you and discuss any potential follow-up studies where this may be relevant. And this is completed with the relevant operational forms. If there are any follow-up studies, then we will go back and provide you with our advice on what you should do in the next stages of your product development. I've just put this slide up to show a typical assay of how we normally work with our clients. We normally work in different stages, and typically we have 3 stages in a full study. You can start at any one of these stages or complete all 3, that's entirely up to you. Stage 1 involves the early identification of adverse events, and this is through our screening assays, which we primarily use T-cell proliferation and multiplex cytokine release assays. These assays normally involve 10 to 20 compounds, and we normally recommend completing each compound in N equals 10 donors per compound. We also have expertise in-house where we can develop specific blood-based or whole blood screening assays for your requirements. And so the services that we can provide here are flexible. This is just an example. If you identify any problem compounds in the screening assay, you may want to understand this fully, and this brings us on to stage 2. This is the identification of the problem compounds using the SCIMMUNE assays. We would normally use the SCIMMUNE platform here, and this may be the small molecule drug platform or the monoclonal antibody, SCIMMUNE mAb. At this stage, we are normally working with 2 to 5 compounds, and again, we recommend using 10 donors per compound in this assay. If you identify problem compounds here, you may want to understand the mechanism of action so that you can redesign your compound or biologic. And we have additional functional assays that we can run along with SCIMMUNE to help you understand this information. We have expertise in multiplex cytokine analysis. If there's an allergic type response, we can set up IgE release assays. We can set up immunohistochemistry assays to understand protein analysis, for example, the heat shock protein 70. And we can also look at the genetic level using gene expression analysis, such as mRNA-seq. At this stage, you are normally looking at 1 or 2 problem compounds. So, in summary, preclinical information on the safety, efficacy, and dose responses to novel compounds can be determined using the SCIMMUNE assays. The SCIMMUNE assays are more physiologically relevant to humans than animal studies, and this has been demonstrated by the Northwick Park trial disaster. Alcyomics assays are human-based, and so are non-animal alternative assays. Both human cells and tissue is incorporated into the SCIMMUNE assay, unlike other human-based in vitro assays. The incorporation of the blood and the skin provides a sensitive assay to correctly identify those drugs which are problem or not. Histopathological changes in the skin tissue are representative of the adverse immune reactions occurring to novel compounds. And SCIMMUNE 3D, the full thickness 3D skin equivalent model, is a representative of normal human skin and can be used as a preclinical screening tool. Alcyomics have a comprehensive portfolio of assays which are reliable, fast, accurate, and cost-effective. Thank you very much for listening to this webinar, which was in association with the Drug Safety Testing Center, the DSTC in Japan. If you have any questions, please type them in the message bar. Alternatively, you can send them after this webinar on the email addresses at the bottom of the screen. We shall now